I'm really excited to be here and see all of you. I've met a lot of you before, and give you an update on what's happening with the genetic registry that so many of you have been helping with and been involved with. So today, my two goals of my talk are to kind of give a genetic framework for how we're thinking about PHACE syndrome at this point, and then give you an update on the current status of the registry. So I thought I'd start by talking about what is a syndrome. So a syndrome is defined as a set of signs or symptoms that appear together that characterize a disease or a medical condition. And the term is used when there's more than one anomaly caused by a single etiology. And then this pattern is seen repeatedly among different individuals. And there are generally some hallmark features and then some less common features. So in PHACE, the hallmark feature is the hemangioma, and then maybe we're coming to understand probably the arterial anomalies. And then some of the less common features are the ventral defects and maybe the eye anomalies. And so for some of you who weren't here yesterday, I just thought I'd provide a little overview. So in PHACE syndrome, we have the brain is affected in many children, the skin is affected with the hemangioma, the arteries, the heart, and then the eye anomalies. And so in genetics, when we want to look for a genetic cause, it's important that we have a really well-defined a really good definition for our syndrome. So as we talked a lot about yesterday, in Houston, Texas in 2008, we had our first research meeting and our first family meeting, and it was really exciting, and now this is the second one, and I hope we keep going with this as we keep learning more in the future. But we established our consensus criteria and came up with a definition for definite PHACE and then possible PHACE. And so definite PHACE has the segmental hemangioma plus one major criteria or two minor criteria. And then possible PHACE has the large hemangioma plus one minor criterion. And just to review, for the structural brain issues that can be the major criteria were the posterior fossa malformations in the back of the brain, and that's the Dandy-Walker complex or cerebellar hypoplasia. And then the minor criteria are hemangiomas in the brain, or a midline anomaly in the brain, or a neuronal migrational disorder. And then for the major heart issues, they mainly relate to the aortic arch, as we talked about yesterday as well. There are a variety of different arch anomalies that we're seeing, the coarctation and also the dilated aortas, and then the aberrant takeoff of the subclavian artery, with or without the vascular ring. And then the minor criteria are right aortic arch or a ventricular septal defect. And then Dr. Hess gave just a beautiful talk yesterday. I wish I could hear that over and over again. But he really defined the different major anomalies that happen in the cerebral arteries in PHACE syndrome. And then the minor criteria would be those persistent embryonic vessels that he talked about. And then the major criteria for the eye are the things that happen in the back of the eye, the persistent fetal vessels, retinal vascular anomalies, problems with the optic nerve. And then the minor anomalies are things that happen in the front of the eye, like the cataracts, or a small eye microphthalmia. And then finally, the ventral midline defects, the sternal cleft and the supraumbilical graphy are major, whereas if the thyroid is not quite in the right place, then that would be considered a minor criteria. And then these are just some pictures of some of the repeated patterns that we see with the ventral or the front of the chest differences. With this is what we call a raphe here, supraumbilical, meaning above the belly button raphe, and that's kind of a fold on the skin that happens commonly in PHACE syndrome. And this is a little skin tag that we've seen a few times in some of the kids. And then some kids will have a larger scar kind of down the chest. So what you're all here to find out is what is the cause of PHACE syndrome, and that's what I want to know too. But the cause is still unknown, and that's the reason for this study. And so a lot of people ask, now why is this syndrome, or how is this syndrome genetic if nobody else in my family has it? And so I thought I would take a step back of how we classically think of genetics, and Mendel was one of the first person who really did a lot of experiments with plants, looking at genetics, and then we still use these principles in human genetics as well. Our human cells contain 46 chromosomes that are in 23 pairs, and each chromosome holds DNA, and the DNA is organized into genes. And any little change in the DNA can lead to the development of a disorder. But what we're learning about more now is that this is just really, really complicated. And so everybody has lots and lots of changes, and those don't necessarily cause a syndrome or a difference. And technology is advancing so rapidly. When I first started working on this, we could look across at only 100 points across the entire genome. And then we advanced, and with Dr. Drolet's help here at Medical College, we ran these chips that look at 1 million points across the genome. And then now we're still working on that, but now we can sequence the entire genome all at once and get every piece of information. So it's really, the technology is just advancing so rapidly. But anyway, so we have the genome, and we have all these chromosomes, and then we have the DNA, and then we have those genes, and then the genes make protein, and then the protein have the functions that kind of instructs the cells how to form. And so there's different ways of looking at genetics. So I think people most commonly think of genetics as hereditary, inherited, transmitted, or transmissible. So you think, am I going to be passing this on, or did I pass it on? But it can also be much more complex than that, or just meaning affected or determined by the genes, but it could be a complex interplay between the genes, and that's also genetic. So everybody wants to know, is there a chance that this condition will occur again in another pregnancy? And I think it's really easy with classic Mendelian genetics to say whether or not there's a risk of transmission for the autosomal dominant, autosomal recessive, and X-linked conditions. And I'll just review those to give you a framework. But we're probably down in the bottom half with the sporadic complex or mosaic, and those are much less predictable. So in autosomal dominant conditions, the condition is expressed in individuals who just have one copy of a gene mutation. And so if a parent has the condition, then the baby has a 50-50 chance of inheriting that condition. And the child has a 50-50 chance then of passing it on to their children. And this is just a picture showing that. So you can see up here that the grandfather has a sensory neural hearing loss with a condition called Warrenberg. And so there's a 50-50 chance of passing it on, and so 50% of the kids ended up getting that gene mutation and having the condition. But it can look a little bit different in different people, even with the same change. So one person might have a hearing loss and then some white hair, and then another person might have some discoloration of the eye plus the white hair, but not have the hearing loss. So even there's variability in families, even with the same genetic change. But then Michael here will have a 50-50 chance of passing it on. And that's pretty straightforward. In autosomal recessive, it's a little less transparent, but because you need 2 copies of the gene change, one from each parent to get the condition. And so when people don't always know that they're carriers for the condition. But there's a 25% chance, or a 1 in 4 chance with each pregnancy of passing the condition on. And so here showing 2 parents who are each carriers but don't show the disease condition. And when they have their children, they could have a child who did not inherit any copies of the gene change. And then they have there's a 25% chance of that, a 50% chance of having children who are carriers, and then a 25% chance that they would each pass on that gene change to a child who would then be affected, who would show the manifestations of the disease. And then X-linked conditions are similar to autosomal dominant. Females the difference is that females have 2 X's, and so they have a 50-50 chance of giving one of those X's to each pregnancy and to each offspring. But the males will not transmit the X to their, males can only pass the change to their daughter because they're always going to give a Y to a son, so they won't be transmitting that change to their son. But in PHACE syndrome, as you've all come to know, it doesn't seem to follow, PHACE does not seem to follow any of these classic patterns, because we have no families that we know of yet where there's multiple family members affected. And so we think that this is more of a sporadic or complex trait. But the caveat that I'll give you is that I have not gathered a lot of family history yet. And so there may be clues out there, and I will be, people in the study will be talking more in the future about just trying to get little clues that might be pointing us in different directions to see if there's any clues out there in the family history. So in sporadic conditions, we don't think that there's generally a risk of recurrence in future pregnancies because we think that it was a change that happened by chance during that pregnancy. But sporadic conditions can have either a genetic or a non-genetic basis. So a sporadic genetic disorder could have happened if there was a small mutation or a small deletion of DNA or a small duplication of DNA that happened while the baby was forming. On the other hand, it might not even be genetic, and it might have been an environmental exposure or some other environmental trigger that happened. And so, as we said, complex traits don't follow classic genetic patterns, but it's possible that it's the interaction of a gene change plus a change in the environment that's all coming together to make this the occurrence of this syndrome. And so what could be happening during development that affects all these different organs? Well, just to briefly touch, because a lot of people did ask about this, what happens in development. Basically, you start with 2 cells, and then the cells keep dividing, and they make a little ball of cells. And at the very beginning, the inner cells are called the endoderm, and they give rise to the digestive organs, the lungs, and the bladder. And then the middle cells are called the mesoderm, and they give rise to the muscles, the skeleton, and the blood. And then there's the outer cells, which are the ectoderm, and they give rise to the nervous system and the skin. So any little error that happened during that very, very complex process could lead to the development of a syndrome. And so we think maybe a gene didn't get turned on or off or got turned on too much at a certain time during development, or maybe there was a change that only happened in some cells as it was dividing, and that we call mosaicism. And this is just a picture kind of showing that, where you had the 2 cells and you had 4 cells, but at this 4-cell stage, 1 cell had a change, and then all the cells that come from that cell will now carry that change. And so that's what we call mosaicism. And so only some tissues in the body are affected. And then which cells might have had the change? A lot of you have heard of neural crest cells and asked about neural crest cells. And neural crest cells are important cells that migrate and form target organs, and they are important in formation of facial structures, and they do give rise to the aortic arch, and they are important in the developing brain as well. And so we do think that it's very possible that neural crest cells are important in PHACE syndrome somehow. But it's very complicated because each cell gives a whole cascade of different genetic signals. So this kind of just shows you this kind of cascade of different cells and different genes that get turned on and off. And so we're as we're trying to find a genetic cause, we're looking through at all of this different cascade of genes to try and see if we can find something in there that might give us some important information. So what's our research strategy? We began enrollment in March of 2006. We have web announcements direct referrals from dermatologists, and then a lot of you just coming forward and contacting me, and I really appreciate that. And we have now subjects from all over the world: Canada, Australia, Spain, Argentina. So what will happen if you enroll in the study? We do consent forms, we review your child's medical history, and then we get a small amount of blood from the child and both parents if possible. And then we collect MRIs and other results and try to put all that information together along with, hopefully now in the future, the family history. And then I take the blood and I extract DNA and then look across the whole genome, like we talked about, the genome-wide analysis. And we did that using these Affymetrix 6.0 chips, and this is what they look like. So the little bit of DNA gets dripped onto there, and then that has a whole bunch of information in it. And what it's looking at are these things called single nucleotide polymorphisms. They're base pair differences, or just little building blocks of the DNA, and we look at all the differences. The difficult part, though, is that the differences aren't necessarily a mutation or a disease-causing problem. There's just a lot of variation. As we learn more and more about the genome, we learn that every individual has a lot of variation. But we're trying to link the patterns that we see with the changes in the syndrome. And so this is just kind of a pyramid looking at how we're sifting through this data. So when we start out, we had 1 million different spots in the genome, and then we found about 10,000 spots where there was a copy number change across all of the chips. And then, so what we want to do is look at which ones haven't been reported as being common areas that are variable in everybody. And there were about 500 of those. And so then we want to say, well, how many are a really big and rare change? And so we're going to pick about 10 of those, and then we're going to look at which genes are there and see if it's any of those neural crest genes that I talked about before. And then that's the gold medal when we figure that out. But then we have to really prove that what we found was real. So we have to use another technique to try and prove that that was really a real change. And then we have to try and get more individuals and see if we can repeat that across different people before we can really say that this is significant and this really means something. And we think, everybody's a little bit different, so it is possible that we have a couple different causes here. And maybe people who have Dandy-Walker have a slightly different change than people who have a coarctation of the aorta. And so we'll be kind of breaking out the different groups and looking at them separately. But a challenge with that is the smaller the groups get, it's not as powerful as if you have 100 people who all have the same thing. And so what is the cause of PHACE syndrome? I can't answer it yet, but at this point we do think there's something genetic that happened during development, but it's an interplay between genetics, development, and the environment probably all coming together. I think as our knowledge and technology advances and as we learn how to understand technology, we'll get a much better understanding of this. So thank you to everybody who participated, and you can always email me if you have questions. And so many people have been involved in this research and have helped out. At the Oregon Clinical Translational Center did a lot of this biostatistics. NOVA has been really helpful. NYU, Graham Bly, I have to mention, and then Alona Frieden really helped me start this project. And now here at MCW, Beth Drolet and Uli Brockle and Kelly Deppie. And we also are collaborating with Bill Dobbins, who's a real expert in Dandy-Walker, who's now at University of Washington. And thank you to HIG and everyone who's been supporting my research through that. And the Society of Pediatric Dermatology and the Dermatology Foundation. And thank you for your attention.